Okay, so now we're going to talk about some protein modifications. So this is post-translational protein modifications. And so we're going to have to talk about modifications of membrane-associated secreted proteins, proteins that are cleaved. We'll talk at the next one about glycoproteins, and now we're going to discuss all these different types. Now these words should be very familiar. Methylation, adding methyl groups, acetylation, adding an acetyl group, replacing the hydrogen on an alcohol. Phosphorylation, kinases add, phosphatases take away. Fatty acid acylation and prenylations to allow for your proteins to interact with lipid bilayers and get embedded into lipid bilayers. And also adding those sulfur groups, disulfide bonds, etc. There's also going to be modification by ubiquitin and ubiquitation. So here is a picture that shows a lot of different things that can happen to your proteins. So of course, all proteins are going to start with your messenger RNA. You're then going to build that protein, initiation, elongation, termination, and you get this protein. Now, the post-translational modification occurs after the protein has been made by your ribosomes. And these can be as simple as phosphorylation, very quick on-off kinase phosphorylase. They could be at the level of glycosylation. So you have N-link glycosylation, so N is in your ER, and you have O-link glycosylation in your Golgi. And these are going to be important to help to make the proteins functional. Some proteins need to have these sugar groups attached to them. Ubiquitation is going to target a protein for degradation or not, as we will discuss. You can have small protein ubiquitation is going to modify the protein instead of targeting it for degradation. We have hydroxyl groups that can get added. We can add a methyl group. Methylation is common in modifications of many different types of proteins. Lipids are going to be important in basically in proteins that need to interact with our lipid layers, prenylation, for example. And adding an acetyl group to the N-terminus is very important. In those lysine, this is very important again for histones that we have discussed in controlling histone activity. So we want to talk a little bit more now about ubiquitation. So ubiquitin is a protein. That's all it is. This is a protein. And so this is going to coat other proteins. And these proteins are then going to either get regulated, silenced for a period of time, deubiquinated, or they're going to feed through a proteasome and get chopped up into pieces that can then be recycled. So to add ubiquitin to an actual targeted protein is going to require 3 enzymes. An enzyme 1, which is referred to as an activating enzyme, an enzyme 2, a conjugating enzyme, conjugation of ubiquitin to the protein, and then enzyme 3, a ligase. And so basically, in the first step, you're going to have your ubiquitin protein, shown here. And then your ubiquitin protein is going to be complexed with enzyme 1. In essence, a monomer of ubiquitin will attach to the ubiquitin activation enzyme 1. And that's shown here. And then this ubiquitin activated enzyme 1 is going to activate, these activated ubiquitins then going to be transferred to the ubiquitin conjugating enzyme 2. So enzyme 2 will swap in and you'll get that enzyme 1 released, and now your enzyme 2, which is your conjugation enzyme, has ubiquitin attached to it. Now your ubiquitin attached to E2 is then going to transfer your ubiquitin to a targeted protein. And enzyme 3 is the lyase transfer. of ubiquitin from E2 to your targeted protein. And this is, of course, associated with lysine, that lysine residue. Now, your targeted protein with ubiquitin can become polyubiquitinated. And so this would have many ubiquitins attached to it through those same steps. E1 grabs the ubiquitin, transfers it to E2. E2 has the ubiquitin activated, transfers it to the targeted protein. Now, the more ubiquitin, the more likely you're going to feed through the proteasome. The less ubiquitin, the more likely you're going to get deubiquitation. And so the process of whether a protein is actually going to be targeted for degradation or is simply going to be modulated and regulated is going to be based on the amount of ubiquitin that's actually added. And so again, you can add ubiquitin to a protein and it does not have to be degraded. It can simply be deubiquitinated, and that will prevent its degradation. So the deubiquitinating proteins are going to be involved in regulation, not degradation. So it's important just to recognize that it was once thought that once you add ubiquitin to a protein, you are going to get it degraded, but that's not true. And so now it's well understood that ubiquitation can just be a simple modification to control the protein rather than to target it for destruction. Now, also, ubiquitation or the inability to add ubiquitin to proteins can lead to disease. So cancer, for example, cancer is for overgrowth, so not being able to degrade proteins that are involved in growth, such as RAS, will potentially lead to cancers. Degrading cell cycle proteins, again, those that promote cell cycle, if you don't degrade them, you can lead to things like cancer again. Degrading those tumor suppressor genes too much, so if you over-degrade certain genes, again, that can lead to cancer. And so it's also been shown that an accumulation of protein in neurons has been linked to Alzheimer's and Parkinson's disease. And one thing to keep in mind is that too much is always going to be a problem, just like too little is going to be a problem. So when you're unable to degrade things, that's just as bad if you accumulate things.